So today we are going to discuss about complementation. What is basically complementation? It is a very common term used in the field of genetics. So what is complementation? It is a genetic test that is used to determine whether the two mutations that cause the same phenotype are present on the same gene or on the different genes. So if the mutations are going to be present on the same gene or on the different genes, and the phenotype which you are going to observe is controlled by how many genes. So these are the two basic questions if you have in your mind, then you can perform the complementation experiment. For example, if you see you have one mice that has a mutation in gene 1, and you have a mice 2 which has a mutation in gene 2. If you are crossing these mice and if the trait is controlled by both of these genes, gene 1 and gene 2, in that case, your phenotype, your progeny will get one copy gene 2 from the mice 1 and one copy gene 1 from the mice V. And because they are getting both copies, in that case, they complement each other, or you can say they belong to the different groups. And when they are complementing each other, it means the mice, the progeny mice, is getting the good copy of both the genes, one from the mother, one from the father, and that is why that is going to be the wild type progeny. So this is the process known as the complementation. So for example, if suppose you have one mice, we just understand this by some example. You have a mice that has a tail, and you have the second mice that also has a tail. Okay. So this is, let's say, mice 1, this is mice 2, and you are crossing them and you are treating them with the mutagens. And by doing several experiments, you are getting one mice out of so many mice that is having no tail. Okay. Now what will you do? And you might get many mice which are having no tail. So let's say you got 4 mice which is not having any tail, and you can say this is the mutant mice and this is the wild type mice. To perform the complementation experiment, it is essential that you have the mutant strain. Without having mutant strain, you cannot perform the complementation experiment. Okay, so let's say you got 4 mice which are mutant and which are, let's say, not having tail. Mice 3, let's say mice 4, mice 5, and then mice 6. So we can write it 4, sorry, 3, 4, 5, and 6. Okay, so what will happen? Let's say we multiply these two mice, 3 and 4, and then we are getting the mice that is having the tail. It means the wild type phenotype is restored. And when we are crossing 3 and 4, we are still getting the mutant mice. It means the wild type phenotype is not restored. So in this condition, these two mice are complementing each other. These two mice are complementing each other. So whenever the mice, when they complement each other, it means they belong to the different complementation group. This is very important point. They belong to the different complementation group because they are complementing each other. Okay, it means, suppose in case of the 3 and 4 mice, if we talk about, you know that progeny will get one copy of the homologous chromosome from the mother and one copy from the father. Let's say this is homologous chromosome from the mother, or this is homologous chromosome from the father. Okay, and these are two mice. And let's suppose or assume that this tail formation is governed by two genes, because you are getting the wild type phenotype. So let's say this is the gene A, this is the gene B, this is the gene A, and this is the gene B. In mice number 3, this gene might be mutated. So this is the region it is not having any tail. Similarly, in the mice 4, maybe the gene B is mutated. So that is the reason the mice 4 is not having any tail. But now see the progeny getting the good copy of this particular A gene from mother. This is fourth mother, I believe. So A gene from the father and good copy of the B gene from the mother. It means both of these genes, when it get this progeny mice, it is having tail. It restored the wild type phenotype. So what does it mean? The tail formation is dependent on the two genes, not on the one gene. Okay, and these genes are present on the different homologous chromosomes. Okay, so now coming to again, if you want to see this, whether the two mutations that cause the same phenotype are on the same gene or the different genes. So in this case, you can see the tail formation is caused because of two genes, not because of single gene. And how many genes are controlling the trait? Two genes are controlling that trait. Okay, so that is the thing which you can see why the complementation experiment is done. Similarly, if you see the mice number 5 and mice number 6, they are not complementing each other. It means they are having either the mutation on the same genes, either in gene A or gene B. If we talk about 5 and 6, let's make it here for 5 and 6. You can say this is 5 and this let's say from mother, this is let's say from father. Both are having the mutation either in the A gene or in the B gene. So the progeny is not getting the two set of good gene in it, means it is not getting the good gene of A in it, not getting. Okay, and we are talking about the progeny, so it is again tailless. So complementation experiment, why it is done? When you want to know when the mutations are present on the same genes or on the different genes, or how many traits are controlling that particular, how many genes are controlling that particular trait. And complementation experiment you can only perform when you have the mutant strain. When you do not have the mutant strain, you cannot perform the complementation experiment. Okay? Now, uh, see, this is the again the good example. How many traits are controlling that particular gene? How many genes are controlling that particular trait? For example, if you see here, you have the 10 mutants. You isolate 10 mutants with the same phenotype. Let's say tail formation only. You need to test every pair. If the mutants fall into three groups that two complement each other between each group, it means the trait requires three different genes. This is called the complementation grouping or complementation map. For example, if you see here, you isolate the mutants A1, A2, B1 and BC1. Let's say these are the four mice you get. You perform the crossing out in each other. A1 to A2, A1 to B1, then A1 to C1 and then B1 to C1. Only these four crosses are possible. In this there is no complementation. It means they belong to the same complementation group. They have the mutation in the same gene, so they are not complementing each other. Now You cross A1 and B1, they are complementing. It means they are on the different gene. B1 and C1, they are on the different genes. A1 and C1, they are on the different genes because they are complementing each other. So you are getting three complementation. So it means this particular trait is going to be controlled by three genes, which is A1, B1, and C1. So there are three genes that is going to control your phenotype. So this is again you can do when you have the mutant strain. Again, you need to remember when the two groups are complementing each other, it means the mutations are present on the different genes or they belong to the different complementation groups. It is like if suppose there is a concert in which only the person who knows singing and who knows dancing is allowed, and suppose you know only singing and no dancing, then you need to find a partner who know dancing also. Then both of you, if you will go together, maybe in the concert they will allow you because you are complementing each other. One is singing and one is dancing. It is like this. So this is regarding the complementation, why it is basically performed. Now coming, uh, complementation is always of two types: cis complementation and the trans complementation. Cis complementation means when the two mutations are present on the same homologous chromosomes. Trans means when they are present on the different homologous chromosomes. So uh, let's see this. This is the single copy of the homologous chromosome. This is the different copy of the. In case of the cis complementation, okay, in case of the cis complementation, whether the mutations are on the same gene or on the different gene, and this is the homologous chromosome from, let's say, a father, and this is the homologous chromosome, let's say, mother. So one gene, two gene, or three genes, it doesn't matter. One, your progeny, this is, let's say, progeny, it will, so the mutations are going to be present only on the one homologous chromosome, but the second homologous chromosome is not going to be affected. So wild type, the progeny will get good copy of all the genes from one of its homologous chromosome or one of its parent. So whenever the mutations are present on the same homologous chromosome, this is known as the cis complementation. Okay? So in this case, you will always get the wild type phenotype, except in the few conditions. And can you think about these conditions? You know the autosomal dominance conditions. When a single copy of the mutant gene is sufficient to cause any disease, in that case you will get the mutant phenotype. Okay. Otherwise, you are going to get the wild type phenotype. And why you are going to get the wild type phenotype? Because the progeny will get good copy of the one homologous chromosome from its one of its parent. So one homologous chromosome is not going to be affected. Only the other chromosome is going to be affected. This is the cis complementation, and in majority of the cases you will get the wild type phenotype because the progeny will get good copy of all the genes from one of its homologous chromosome or from, or you can say, homologous chromosome of one of its parent. So this is a cis complementation. There are some exceptional cases which I can tell you here. There is a haploinsufficiency. When the one wild type copy is not enough, then a mutation in cis behaves dominantly. In that case, you can get the mutant phenotype. Dominant conditions I already told you. If the condition is like autosomal dominant, then single copy of the dominant gene is enough. Single copy of the mutant gene is enough to cause any disease. So in that case, you will get the mutant phenotype. And the gain of function mutation. Mutant allele produces new activity and it becomes dominant in that condition. So these are the three conditions where in case of the cis complementation also you will get the mutant phenotype. Otherwise, in majority of the cases you will are going to get the wild type phenotype. Now coming to the uh, trans complementation. So trans-complementation mutations are present on the different homologous chromosomes. So let's say if this is two genes that are controlling one trait. So let's say this is A, this is B, this is again A, this is B. This is from the, let's say, father, this is from the mother, and this is the progeny. Then then the mutation could present on the different chromosomes. So let's say in this or in this, if this is the case, the progeny is getting good copy of A gene from female partner and I mean its mother, and the good copy of B gene from its father, it is going to be the wild type. But what happened in case of the complementation also, if suppose there are two genes A and B, mutations are present on the different chromosomes but on the same gene, then in that case this is going to be the mutant phenotype because A gene is defective in both the chromosomes. So, you know, in the father also A gene is defective, in the mother also A gene is defective, and the progeny is getting the A gene is defective A gene and only getting the good copy of B gene. And because the trait is controlled by both the genes, the progeny is going to be the mutant phenotype. It could happen in case of the single gene also. In case of the single gene, if you see, if this is your progeny and the single gene is controlling particular trait, then if the mutations are present on the same gene also, you are always going to get the mutant phenotype. Okay, so this is like the trans complementation. So that's all about today's lecture. Thank you